Noel is a master. He's our videographer in residence, and he's doing all of our, you know. Alrighty, first and foremost, I want to thank you all for coming. It's really exciting for me in particular, because this bio lab is a new feature for CircuitLaunch. And it's exciting for me to kind of build our community, especially since we built a really wonderful community around robotics and hardware. And as I've told some of you here, there are similar challenges, people in biotech, as there are in robotics and hardware. And we're really excited about trying to build up our life science community here, as well as support the rest. And so I also want to thank Shane, Joan, and Doug from the bioinformatics group. They've been really integral in helping me kind of jump into the fire here. So I'm going to leave it to Shane. And so first, we're going to have a couple speakers, and then we're going to have a panel, and then we'll have a little bit of networking at the end. And if you would like, I can give a tour to anyone who'd like to check out the facility, check out our lab facilities, and of course, if there's any questions, we can always do that as well. So I'll leave it to here, Shane.
Thank you. Thanks, Patrick, for having us. Yeah, thank you everyone for coming. There will also be a little short break for announcements towards the end. So if you have a job opening or things like that, please get ready and you can come up for 1 minute and give your announcement. So first I'd like to introduce our keynote speaker, Peter Karp, a amazing and preeminent bioinformatician from SRI, and he's going to be talking to us about some really cool new tools and how to expand the paradigm of microbial genome annotation. Thank you, Peter. So you can stand here.
Sounds good.
Yeah. And you can,
I'd actually like to sit. Is that going to work out?
sure.
For you?
I think you could sit here. And you can use this, or you can have that next to you.
This one? Or?
Yeah.
That should be fine.
Yeah.
Let's give it a try.
Try that.
Well, thank you so much, Shane, for inviting me, and nice to reconnect with this group. It's been a number of years.
Is this you?
Sorry?
This is you?
It is.
Yeah. Okay. So you can use the clicker to advance.
Okay.
Anytime you'd like. The right arrow takes you forward. The left arrow takes you back.
Great.
Wonderful. Thank you.
Let me just quickly introduce where I work, which is SRI International. And as I was talking with a few of you, there are at least 3 SRIs, 3 research SRIs around the country. But ours used to stand for Stanford Research Institute. But Stanford became independent from SRI in the 1970s. So SRI doesn't stand for anything anymore. We also just announced that we're actually merging with Xerox PARC sometime very soon. So we're all kind of wondering if the name is going to change, and I haven't heard anything about that yet. But we're a nonprofit research institute down in Menlo Park with a number of offices around the country, but now soon to be a new office on Coyote Hill Road. And I thought what I would do in this talk is it'll have a significant historical component where I'll look at how microbial genome annotation in general, has changed over the years. How has the field matured? Maybe what would people have thought 30 years ago when the Human Genome Project first started? How would they look now at the genome annotation systems that we use now? So we'll look at what were some of the, I can't read that one, so I'm going to have to turn my head a bit here. What were some of the early predictions of how microbial genome annotation would work? We'll take a look at that. What is the standard paradigm of microbial genome annotation today? One thing I realized is I think most people have a pretty fixed idea of what we mean when we say genome annotation, but in fact, the field has matured quite a bit, and there are actually many new layers of information that we figured out how to add during the genome annotation process that I think have expanded the paradigm significantly beyond what people typically think of as microbial genome annotation. I'll talk about some of the annotation tools we've developed in the Pathway Tools software and BioPsych, both of which my group has been developing for many years. And I'll talk about a new tool that we just released on the site recently called the Comparative Genome Dashboard. Okay. Yeah, and one thing I'll note is that although the Human Genome Project itself had a very clear holy grail, namely obtaining the human genome sequence, I don't think there was ever any really clear goals set up for the annotation part of it. You know, there wasn't any clear holy grail, and so we can't really assess how the genome annotation field has performed against a holy grail because there wasn't one, but we can still look at some of the early expectations. And so I found this publication from 1988, back when I was still a graduate student, and found a couple interesting quotes in here about how people thought the human genome would be annotated. And so here's a nice quote. The benefits of the Human Genome Project are the benefits that will accrue from organization and scale to carry out in one place or a few places the work that otherwise will be done in fragmented form over and over again in individual laboratories. The product will be a database that will contain the human sequence and all the information known about it. The database will contain initially, as one analyzes the sequence, putative candidates for genes. It will contain computer identifications of possible structures on the DNA that can code for proteins and suggestions of what those proteins look like in terms of their primary amino acid sequence. And if I remember, we're aiming for 45 minutes here. Am I right?
I think that's fine.
Okay. It will become a reference database which one would use to plan future experiments and analyses. This is from Walter Gilbert's chapter in that book that I just put up. And so here's a figure from his same chapter. If we have a genome database, new science will emerge, will recognize patterns and predict genes and their controls. And then he talks about the structure-function problem and says we're going to go from a DNA sequence to an amino acid sequence to a 3D form to a function. And that'll be solved in 5 or 10 years. Well, maybe 30. And maybe we can go to the 3D form now, but I'm not sure we can go to the function. I'd be curious other people's thoughts on that, but maybe for a small fraction of the proteins. But so they were a little off there. But it was interesting to me that in 88, I would have thought sequence analysis would have been put forward as the main tool for predicting function. But surprisingly not. I mean, surely Smith-Waterman existed then. BLAST, I think, was just coming out around that time. So overall, the expectations were pretty low back then. They identified gene finding as an important problem. They identified predicting gene function as an important problem, and predicting control regions as another important problem. And I think here's another challenge that they could have stated, which is, given a genome sequence, automatically generate a computational model of the organism. I think if anyone had stated that as a goal back in 1988, they probably would have been locked up, put in a padded cell. But in fact, we've largely achieved that. At least we can now create metabolic models from a genome sequence, which I think is a pretty amazing achievement that's gone way beyond what was foreseen back then. And we'll talk more about that in a few minutes. So here's what I see as the standard paradigm of genome annotation now. You take your reads off your sequencer, you assemble them into larger contigs, you run gene finding programs to find where the genes are, and then you use tools like BLAST and HMM searches to predict the functions of those genes. I think that's what most people think of as the standard paradigm of genome annotation. And it's implemented now by some of the standard tools for microbial genome annotation are PROCA and RAST and the NCBI prokaryotic annotation pipeline. There have been a number of other systems developed over time, like MagPie and GeneQuiz, a few I can't remember the names, that have all kind of fallen by the wayside. But these are some of the tools that have remained. And here's what I see as now the expanded paradigm of genome annotation, where These additional steps have gone pretty far beyond that standard paradigm, and these additional steps include predicting the metabolic reactions of the organism and the metabolic pathways of the organism, predicting what we call pathway hole fillers, or which genes in the genome code for enzymes that are missing from metabolic pathways. Because when you do pathway analyses of most genomes, there are always missing enzymes in some of the pathways because they're hiding in the unannotated genes. Predicting transport reactions, generating a metabolic model using methods like flux balance analysis, predicting the operons of the organism, and predicting the protein complexes of the organism. So our Pathway Tools software actually does all of these additional steps, 4 through 10. So when we ingest the genome, our software does not do steps 1 to 3. We assume that's already been done by an external software package like the ones I just listed. And then there are other packages that do some of these other steps, KEGG, Model Seed, KBase. But yeah, that's what I mean by the expanded paradigm. And so actually you can actually look at genome annotation from another point of view, and you can say, well, genome sequencing isn't a goal in itself, right? Really, the reason we sequence a genome is to learn more about the organism. And so really our goal is to get as much information about the organism as we can. And so to most efficiently construct a comprehensive knowledge base for a given organism, we'd want to combine the genome sequence with computational inferences applied to that sequence, with data imported from other sources, and with manual literature-based curation to supplement all those other forms of information. And so this is yet another expanded notion of genome annotation, or of creating a knowledge base for an organism. And so this is the approach that BioCyte uses for annotating genomes and creating new pathway genome databases in BioCyte. We, let's see, is there a pointer? Yeah. Can you see the pointer on the screen? Probably not. Okay. So we really have 3 tiers of methods that we apply to a new genome that we're adding to BioCyte. We perform this whole set of computational inferences. Really, it's the same set that I showed on the previous slide, predicting metabolism and operons, protein complexes. We perform data import from multiple different sources, from UniProt to regtransbase. And then for some of the organisms, a selected subset, we perform literature-based curation, which involves correcting a lot of the computational inferences and adding in information from the literature that's been determined experimentally for that organism. So the BioCyte database collection, we've now followed that scheme for 20,000 different genomes, of which 69 are curated. And those 69 databases are curated from 130,000 publications, of which for E. coli alone, we've curated 43,000 publications. The BioCyte website supports these 5 main different modes of use. Information seeking, seeking out information about the function of a given gene or a given pathway or a given reaction. Analyzing high throughput data sets, including transcriptomics and metabolomics data. We have a number of comparative tools. I'll talk about a new one tonight. And metabolic engineering, synthetic biology is another big use case for our tools. The BioCyte website is powered by a software toolkit called Pathway Tools that lets you both install a private set of these existing biopsych databases as well as run new genomes through the software. So Pathway Tools is the software that does all these computational inferences on the top. And so at biopsych, we've applied those inferences to a certain set of genomes, but other groups like to run their own genomes through it to create pathway genome databases for their own genome data. And the software provides an enterprise pathway genome data management environment, lets users interactively edit PGDBs to add new gene functions and pathways. It supports construction of quantitative metabolic flux models using flux balance analysis. And it supports these same use cases as biopsych does. And here's the basic use scheme for Pathway Tools is you input an annotated genome, annotated using those first 3 standard techniques. Then it applies those extended set of techniques, 4 through 10, to create a new pathway genome database, which then can be operated on by these other modules of the software. The Navigator software that supports search and visualization and omics data analysis and comparative analysis. The editor tools that let users interactively edit the data, and the MetaFlux tool that supports creation of metabolic flux models. This is a summary of the overall capabilities present in Pathway Tools, and I'm not going to read through every item on the list, but I'll just note that they're divided into genome informatics and pathway informatics, regulatory informatics. There are a number of microbiome capabilities, a number of comparative analysis capabilities, transcriptomics and metabolomics analysis capabilities, and what we call advanced data manipulation, ranging from APIs to a tool we call Smart Tables, which is kind of like spreadsheets driven by a database. And within these tools, you'll see some commonalities, like a number of them will have search tools and information tools, information pages geared toward that type of information. So there are reaction and metabolite and pathway search tools and information pages. There are zoomable metabolic network diagrams. There's a tool for interactively exploring a metabolic network, starting at one metabolite and then moving forward or backward through the network, one reaction at a time.
What is PGDBs?
Sorry, Pathway Genome Database. So that's just the term that we use for the kind of database that our software creates. Yeah, and so when we create a PGDB, our software can use those databases in many different ways. We can mount them on the web, which is when you're interacting with biopsych, you're essentially getting a web view of the database. You can search the database using many different query tools that we've developed. You can create a zoomable metabolic map from the database and create a metabolic model from the database. I think I'll skip this slide. I'm not going to get into how metabolic reconstruction works in that much detail. Well, I'll kind of summarize how metabolic pathway inference works. It starts with our Metapsych database, which is a database of 3,000 metabolic pathways from all domains of life that we've curated from the literature, curated from 75,000 publications. And the way we predict metabolic pathways in a new sequenced genome is for each pathway in the Metapsych database, we compute a score expressing the likelihood that that pathway is present in a given genome. Could be a microbial genome, could be a eukaryotic genome, whatever. And here are some of the main features that scoring is driven off of. Essentially, it's looking at reactions inferred by enzymes in the genome. And so there's a previous step where for each enzyme in the genome, we look it up in Metapsych and predict what reaction that enzyme catalyzes. So we then get the reactome of the organism. And so now to score the pathways, we look at each reaction in the pathway and ask how many of those reactions have an enzyme present in the genome. Maybe the pathway is complete, an enzyme is present for every reaction in the genome, maybe there are just a few missing, or maybe none of them are there, or just one or two. We also look for each of those reactions, how many other pathways that it's in. In other words, is the reaction a unique signature for a given pathway, or is it really present in so many pathways that it doesn't add much evidence to any one given pathway? We also ask if a given pathway is outside its taxonomic range. That is, in Metapsych, we specify, oh, this pathway is a plant pathway, this pathway is likely to be a bacterial pathway. So if we're analyzing the bacterial genome, we require a really strong score for a plant pathway to be predicted into a bacterial genome. We also specify in Metapsych key reactions for the pathway, such that if we don't have enzymes for the key reactions, the pathway will not be predicted. And this helps us distinguish between pathways that have a lot of similarity, like the Calvin cycle and the TCA cycle, share a lot of enzymes in common. And so this will help us distinguish which of those two pathways is present. And we also have a notion of variant pathways. For example, there are 9 different versions of the TCA cycle present in Metapsych. Oh, thanks. And so we have some special rules to try to figure out which of those variant pathways is present in a given genome. I think I'll skip that one. And I'm going to skip that one. So let's talk about operon prediction. So since 2004, we've had an operon predictor within Pathway Tools, and I thought I'd give you a little update on that predictor because we've revamped it recently and made some improvements to it. The basic approach in both the early version and the version we have now, the new version, is to look at every adjacent pair of genes in the microbial genome and ask 2 things. We essentially want to compute, are A and B within the same operon, or are they at a boundary between operons? Okay, so that's what we're trying to determine. And how do we determine? And in this diagram, the 3 genes in orange are within the same operon, so like that's a within operon pair. And here we have a boundary between our orange operon and our blue operon. So the previous approach we used considered these aspects of the 2 adjacent genes to compute whether they're at a boundary or within the same operon. We looked at transcription direction because, of course, genes that are not in the same, transcribed in the same direction are not going to be in the same operon. We look at intragenic distance because that's a really strong, that's the strongest signal for presence in the same operon. And we look at whether A and B are functionally related in some way. Because as we know, genes within operons often are functionally related, and that's why the cell wants to express them together. And in the original version of our predictor, these are the tests that we used to determine if 2 genes are functionally related. We looked at whether they're in the same metabolic pathway. We looked at whether the first gene codes for an enzyme in a pathway and the second gene codes for a transporter involving a substrate in that pathway. We looked at whether A and B are subunits of the same protein complex. And using that older predictor, we were able to get an accuracy of 79.6% when evaluated on experimentally determined E. coli operons from our EcoCyte database. So we made several changes to update the operon predictor because that operon predictor has become out of date, and we had some new ideas we wanted to try out. Oh, the old one also used the multi-fun ontology as another related way of evaluating functional relatedness. That was an early precursor for gene ontology, which is now really obsolete. So one of the things we did was to take out the multi-fun component and add in gene ontology. So should be here somewhere, yeah. So if A and B are in the same gene ontology biological process, we'll also say they're functionally related. Some of the other new features that we evaluated were, what if we have some gene expression data for this organism, and A and B have correlated gene expression profiles? In that case, we'll also say A and B are functionally related. So here's a funny one that really people in this field have missed for 30 years, including me, but incredibly obvious. If the genes begin with the same 3 letters, they're probably functionally related, right? Because that's how people name bacterial genes. So that's another measure of functional relatedness. Now it's true that for a long time, some of the annotation pipelines would not assign gene names, like the NCBI PGAP would not assign gene names, but they've fixed that. Also, we've also found that NCBI now has many more gene ontology terms present in its annotations. Just in the last couple of years, they've improved that a lot. We also looked at things like, do A and B have the same product type? Do they have similar GC content or similar codon usage? And even do they have similar length? Just under the idea that maybe if they had radically different lengths, they might be doing very different things. So all of these features had a little bit of value, and none of them really improved accuracy that much. So our accuracy went from 79.6% to 83%. That's the best we've been able to do so far. But that is a 17% decrease in the error rate. And like it turns out, the gene expression and the similar gene names were the most informative features, but they just didn't add that much beyond the features we already had, which is a little surprising because, for example, I would think the gene name in particular, I would think both of them would be orthogonal to like looking at pathways that we were doing before. But for whatever reason, we didn't get that big of an increase in accuracy. It's not going forward. Can you give me a next slide, or? Thank you. So this is another new predictor that we've just built, and what this predictor does is its input is the proteome of some organism, and its output is a grouping of proteins from that proteome into protein complexes. And the approach is, it actually uses 3 different techniques. The first is it does a syntactic analysis of the names of all the monomers in the proteome, which I'll describe in the next slide. It also looks at monomers that catalyze the same metabolic reaction and share certain keywords like subunit or chain and are proximal on the genome, and if it finds monomers that satisfy those conditions, it infers that they're part of the same complex. And finally, it looks at monomers in the same operon that have, again, names, words in their name that suggest they're part of an ABC transporter, in which case they're probably part of a complex as well. And here's the first approach, the syntactic similarity, is we look for proteins in the proteome whose name includes a subunit word, like subunit or chain or monomer. And we strip off all these subunit words to generate what we call a core protein name. And we find proteins that have the same core protein name. So it's based on the very simple assumption that if you look at these 2 protein names, you're going to infer that they're part of the same complex. And so that's really what this software does. It's not using any fancy 3D structure analysis to determine if the proteins bind together. It's simply using an analysis of their names to infer what the complex would be. So here's another way in which I think we've extended the genome annotation pipeline, is to find genes that aren't in the genome, but should be, or what I'll call missing in action genes. And put another way, it's identifying genes, functions that have to be hiding somewhere within the genome. And we have 3 different ways of doing this. The first one is one of the slides I skipped. I think I mentioned the notion of pathway holes, or reactions within predicted metabolic pathways that have no enzyme assigned to them. And probably those enzymes are hiding somewhere in the genome. There's also a notion of reaction gaps. It sounds the same, but it's a little different. And these are reactions that are missing from the genome that when you go to build a flux balance analysis metabolic model for the organism, ultimately, if you're going to have a working metabolic model, you need to have a path from the nutrients of the organism to the biomass metabolites that its metabolism is producing. If you don't have a path because of missing reactions, you won't have a working metabolic model. And there's a technique called gap filling, where basically it's a software tool that tries to bring in a minimal number of reactions from a database like Metapsych, a reference universal reaction database, tries to bring in a minimal number of reactions to your metabolic model that will enable all the biomass metabolites to be produced from all the nutrients. And so this is another way. The reactions that are postulated by the gap filler are another kind of missing in action protein. Those functions are probably hiding in the genome somewhere, but they haven't been found yet.
Peter, I'm sorry, can I just adjust your mic for a second?
Sure.
Thank you. Sorry, folks. I just want to get the best sound possible by putting this right here.
Sure.
Thank you, sir.
Thank you. And so a similar situation applies actually for the protein complex predictor, that when you run it, okay, you often find pairs of proteins or triplets or quadruplets that have the same core protein name, but you also find singletons, right? You might just find the alpha chain and no beta chain. And so that's another kind of missing in action protein, that probably that beta chain is hiding somewhere in the genome and you haven't found it yet. So, actually, before I do my summary slide, I'll show you this new other tool that I mentioned called the Comparative Genome Dashboard. And this is a tool where its input is one or more pathway genome databases for organisms that you want to compare. And what the tool does is to give you an interactive dashboard that lets you visualize all the functional subsystems within the cell, and then drill down within different subsystems for more information. So each of these panels is one set of subsystems. So this is the biosynthetic machinery of the cells, the degradation machinery of the cell, transport machinery, response to stimulus, central dogma, and energy metabolism and precursor metabolites. Those are the main systems. And then we have all these different subsystems underneath them. So under degradation, we have amino acid degradation, nucleotide degradation, fatty acid lipid degradation. And so what we're seeing, what the different bars represent is, actually the bars mean 2 different things depending on which panel you're looking at. In these panels, they refer to the number of compounds. So they refer to the number of amino acids that can be synthesized, nucleotides that can be synthesized, or amino acids that can be degraded. And if we click on any of these bars, we'll get a new window that lists all the different nucleotides that can be degraded or synthesized. Now, for this one, for example, The y-axis is actually the number of gene products involved in that subsystem. So this one is starvation, heat shock, cold shock, DNA damage. This is showing us how many different proteins are involved in that area of the cell. And again, if you click on it, you'll get a list of all the specific genes. So this is a way of looking really at a glance at what different functional subsystems are present in a cell, how much of the cell's machinery is it devoting to it. If you're comparing 20 different bacteria and you want to find which ones can make proline, you can find that out in about 2 seconds by clicking on that particular one. And all this information is based on, well, the information that populates this diagram is coming from 2 sources. It's coming from the pathways in the pathway genome database. So in other words, when we generate our list of amino acids here, we're looking at which amino acid biosynthetic pathways are present in the PGDB and extracting the actual amino acids they make, and that's what populates the list of compounds here. For these, most of these are generated from the gene ontology annotations, and that's actually part of the reason why these are the blue ones are totally missing. The blue genome is missing because it has no gene ontology annotations. So thank goodness NCBI is cranking out more gene ontology annotations, because that's going to make this tool work a lot better. Okay, so back to my summary. So I think the genome annotation field has advanced way beyond its initial conception, especially with the ability to ultimately generate metabolic models from sequenced genomes. I talked about what I think most people think of as the standard paradigm, and then I'm arguing that we've really expanded that paradigm in many ways over the years. I've also talked about the notion of automatically flagging the missing elements of a genome annotation, the missing in action genes. And then I've talked about pathway tools in biopsych as an engine for storing and searching and visualizing, analyzing, comparing these extended genome annotations. And I think, yeah, so let me thank my coworkers at SRI and various biopsych and ecopsych collaborators and our funding sources at the NIH. And happy to take any other questions if you have some.
I really like the way.
I'm sorry, could you say it to the mic so the stream can hear us?
The way it flags missing items seems really cool to me, because if you're building a model, you want to do iterative, like improvement on the model, right? And so that's like, it makes it into a cycle, because somebody could then look at and try to find the thing, right? And even if the model itself can't find it, like a human could try to find it.
That's right.
If you wanted to make a whole computational model of an organism, you'd have to do a lot more iteration on a lot more different aspects. So I'm just kind of curious, what are your thoughts on like, how could you have, whether it's automated or human involved, iterative refinements that you could actually get to, like, instead of like 83%, like 99%, like how would that work?
Yeah, I mean, a lot of the databases in biopsych are purely computationally created with none of this iteration, but that's really what our curation procedure involves for those 70 curated databases, is looking for some of these missing elements, going to the literature and filling in missing information from the literature. There are a lot of errors that the curators can spot just by noticing, often the sequence analysis will call a wrong function because it gets a different member of a protein family that doesn't belong in this organism, because the organism doesn't do that. I think searching for missing enzymes has been the thing that I've seen people work on the most. So we have a tool in Pathway Tools called the Pathway Hole Filler. It basically will, given a missing enzyme, it will retrieve from UniProt all the sequences that have that function and blast them against the genome, looking for hits from other proteins. let's see. So yeah, I guess those are the main ideas that I've seen around for doing the kind of iteration that you suggest.
Excuse me. Thanks for the talk. I'm curious. You talked about kind of the initial paradigm, sort of taking short reads, generating contigs, and flowing down from that. I'm curious to what extent you think newer sequencing technologies, say like long reads or the like play into this process, if at all, or how that might change things.
Yeah, I don't really usually get that low in the data analysis pipeline. So I'm not really an expert on assembly and so forth and how the long reads are going to help. So not much to say.
Yeah.
Maybe somebody else in the audience knows more about that.
Hi, I'm Joan. I used to work at PacBio on microbial genome assembly and annotation. So your question was about whether the long reads would change like the annotation or what, anything in particular?
Yeah, I mean, just to what extent, like, the ability to generate microphone, please. The extent to which long reads might percolate down the workflow that Peter was discussing and perhaps make, say, the 83% go even higher or improve, basically improve what he was discussing.
I think I can speculate a little bit on that. Well, first of all, if you have long reads, you may not even need assembly necessarily because you could just get something like 10, 15, even 20 KB that's already like more than 99.9% accurate. So you could predict a whole operon from there, maybe a whole pathway, and it would be accurate. So where I see problems in short read assemblies would be that if you have very closely related members of a gene family, that assembly is not going to be accurate, most likely not going to be accurate because like there's no way to tell whether one repeat is in another. So I think that will complicate some of the predictions of genes and some of the things they might be predicted as pseudogenes or some other artifact when in fact it's just like a variant. I think. Go ahead, Doug.
I was just going to ask, you have a lot of these automatically annotated genomes. Do you have any sort of threshold in terms of sequence quality? And what do you use to measure that before you decide to include it or not? Because I think that speaks to the effect of long reads increasing your sequence quality, but how important is that?
Yeah, we don't use it sequence quality itself as the cutoff, but we do look at simply the number of genes that have predicted functions. Mm-hmm. And there are some genomes out there that have hardly any genes that actually have a reasonable predicted function. So we will cut out genomes based on that criterion. Right. And similarly, when we run the genome through pathway prediction, we require a certain number of pathways per 1,000 genes. And again, if we're getting a really low number of pathways, we figure the genome annotation is probably pretty bad. I don't remember what the numbers are that we use.
Mm-hmm. So in the early days of genome annotation, we used to say if you were lucky, you might get 45% of the genes, like in E. coli, would have a reasonable annotation. Do you think that's gone up since then? What would you put it at these days?
I think so. I mean, it depends on which phylogenetic group the organism is from. Mm-hmm. I think that's one of the dirty secrets of genome annotation is we don't really know how accuracy has improved in the field over time. There are not good benchmarks for comparing two genome annotation systems, and we can't really say how much annotation quality has improved in the past 10 years.
Okay. Yeah, I'd agree.
Unfortunately.
One more question?
Yeah. Great talk, and I think this is really amazing work that's being done on kind of the microbial level. A lot of my work is being done on more complex organisms like insects, mammals, and plants, and these kinds of tools would be pretty amazing if we could, apply them to some of these more complex systems. I was just curious if you could speak to, how easily it is to port, from, these tools that are developed for these microbes over to something like a plant or mammals.
Yeah, I mean, these tools have been applied to excuse me, all the other groups. Obviously, you don't turn on the operon predictor. But let's see. Yeah, I mean, overall, pathway prediction does work in the other organisms. Metapsych does have many pathways from plants and from animals and from insects. We do have at Biopsych, we have a human, mouse, rat, yeast, fly, worm, Arabidopsis. And if you look at plantpsych.org, they've run something like 150 plant genomes through pathway tools. And that's the basis of their website. So yeah, the tools can be applied to many other groups besides microbes.
Thank you, Peter.
You're welcome, thank you.
All right, so next we have Eyal Kiva from Shiru, and he's going to tell us about AI-driven methods for discovering food ingredients. Is this on?
Peter, you got the mic?
Huh?
Oh, that's perfect. Yeah, just put it in your pocket.
I
on
don't
this
have
side
one.
if you're going to turn toward the screen.
Yeah,
You got it?
no pocket, so help me here.
Yeah.
All right. So it's really exciting to be here. Yeah, thank you for the invitation and the awesome venue. So I'm Eyal Kiva. I'm a staff bioinformatics scientist at Shiru. And just before I'm going to tell you about the villain in my story, I want to take you for a ride in the supermarket that I did like the beginning of the week. And since I've been working at Shiru, I developed this obsession about ingredient lists. So whenever I go, I don't take pictures of all of them, but I am interested, especially at the end of these lists. There are all these like, this product contains less than 2% off, and then a list of long words that doesn't sound like food. And many components that are like out there, and even though these are like food ingredients that are in minute quantities, they turn out to be a very key component for very impactful tasks. And I'm going to take you from the supermarket to the world and talk about sustainability. And essentially, when you look at, especially in the livestock production of protein for people, i.e. the food, most of the food we eat. Numbers like 70% of the farmland pastures are used for either grazing or feeding livestock for the meat industry, while only 18% of the calories we ingest are from this type of foods. And the gas emission, greenhouse gas emissions of producing either cows, cattle, but also cheese and eggs is significant. And obviously one of the solutions is trying to change the way, the things that people eat. And ingredients can help us fine-tune any plant product into something that has like some more recognizable mouthfeel and reconstitute the taste that we are looking for. And the market is there. If we look at the massive expansion in the market size from 29 billion for like plant-based market in 2020. 2021 it's around 41. And it's expected to rise significantly and capture a big share of the market. One thing that needs to be taken into account is the issue of market acceptance, massive market acceptance, and people people here will go and buy these like new foods and try them because they are new and they want to eat them, but then you have this cliff. If it's not tasty, the chances of buying it again diminish very quickly, and if it's too expensive as well. So this requires a lot of like fine-tuning and getting to the taste and like approaching the clients, the consumers, so they will be persuaded to buy more and more. So that's, and ingredients are key component in fine-tuning these types of food. And there's a history already, starting with the first generation that had some limitations in terms of both like taste and texture and price and the clean label. And then other more like second generation, a massive improvement in terms of taste and texture. My daughter is a vegetarian. She does not eat Beyond Meat burgers because it's too reminiscent of meat. So that's like, at least for my single point of statistics at home, I know that like there is definitely improvement in taste-wise, but still clean label and price points still have like a place to. You want to ask a question?
What does price or clean label mean?
Clean label is if you have a food product with a GMO label on it, many people will not eat it.
So clean label, no notification of GMO.
GMO, and there are other tags that might, big companies will refrain from integrating an ingredient that is associated with like negative perception by consumers. So that's so we want tasty food with the right texture that mimics the mouthfeel of meat, of livestock-based ingredient, like food, clean label, and a price point. So many types of optimizations to do, and not unfortunately, like livestock food product had around, I would say, 10,000 years of evolution. People, and it's a whole culture. People know their food, and they are it's a very cultural thing. So trying to fine-tune these tastes for clients, like consumers' acceptance, is a big challenge. Yeah. So if I want to take you through one bite of this burger, and obviously, when you look at it, you go through the bun and the cheese and the burger and the sauce and all of them. When you try to dissect them into different features, there are many features, and many of them are owing to different like ingredients. So you want like to imitate the mouthfeel, and you need the gelation agents to structure the fat and the water inside the product, and you need to sometimes emulsifiers to get the sauce thick and creamy. So many functional requirements. And the big thing is like how to find plant-based proteins that can be used to do this stuff, these functional things. And well, I've been many years in academia, and you don't have lots of like, you have the strive to find the function of proteins, but the native function. The function of a protein in a food product is not a natural use of these components. That's why it's like such a great question to try to ask as a scientist. You don't find the answers in many publications. So that's a cool challenge. So in order to accommodate and try to find these proteins, so Shiru came up with Flourish, which is like an end-to-end solution to this type of problem that starts from a protein that I think should be a good like gelation or emulsifying agent, and will end up being tested in the lab in a beef patty and still maintain the shape of the beef patty instead of all these ingredients that are used now in plant-based meats and are not necessarily food. Sometimes they are, for instance, byproducts of the wood industry. They are edible, they're not allergenic, but they go through us the same shape that they're not food. But still very used in many types of food products. So it starts with identifying the target, high throughput screening, meaning taking the gene, cloning it, getting like minute amounts and trying to test it with all sorts of proxy tests, and eventually fermenting it, producing it in high amounts, putting it into patties, and just verify that they do what we thought they will do. And that's and there are all sorts of filters along the way. The protein needs to be expressed, needs to be soluble, and also do the function. So there are many, many things along the way. And one of the things that is like unique when you like traditional ways of formulating food are essentially bound to the raw materials that you can buy, that are commercially available. You buy like some soy and you integrate it with some commercially available ingredients, and you process it, and then you try to all this matrix of endless possibilities to mix these ingredients in order to reconstitute a burger, for instance. And in many cases, and like I worked for so many years on sequence similarity and structure similarity. When you look at an egg, the proteins you have in an egg, and the proteins you have in egg replacements, there's no sequence similarity at all. Like these come from chickens, the other ones come from plants. How do you find the functional equivalence? It's a very tough challenge, but we, like one of the components in Flourish is a special proprietary grouping algorithm that groups together proteins in a very like surprising and pleasantly surprising functional similarity, even though they are not similar in terms of sequence and structure. After a set of some filters, these proteins go from the more digital search and of the discovery into the more produce and test part of Flourish, meaning taking the gene, cloning it in a host, producing small amounts and using high throughput methodologies to test it, soluble, expressed, and some proxy tests, and eventually also fermentation. And this small GIF represents testing it in food, so like maintain some sort of like the function that we are after. And obviously there is this big green arrow that goes back because this system learns and iterates and improves itself with time. So I want to like take one test case of gelation. Gelation is a function that is very important for plant-based meat. Holding the water or holding the fat, or things like one of the when you think about omelets, the fact that you take an egg and you heat it up and becomes more firm is from material perspective a non-trivial thing, because when you heat up things they become more soft. That's gelation, and that's one of the and the question is how to find other plant-based materials that will do the same thing. So methyl cellulose is one of the components that actually is a gelation agent, and it's used for other things. This is a component that is a byproduct of the wood industry, used in many of the pictures I took of plant-based meat. And we are working on a methyl cellulose replacement, a protein that replaces this function. And this small picture represents what summarizes what I found in the literature about the mechanisms of gelation. So we have a protein, and essentially it self-assembles into some sort of fibers and a network that holds the water, and it happens upon heating. And the pictures I found are equivalent to this picture because this is how they look like on the left side. When I just search for like gelation, so I have this picture and that picture, all of them represent exactly the same phenomenon, and they are the mental models of like researchers that try to understand what's happening in this like material that creates the network and holds the water. And we need a better understanding. We need a better model that goes from sequence to structure to cloning to high throughput to fermentation and to a beef patty and eventually a burger. but this is obviously not a very trivial task because when people add some lemon, put some spices, and they sizzle it on a pan, this is like the protein needs to be temperature tolerant and pH tolerant and ionic strength tolerant. So you have like many things to predict here, and in order to find the right sequence, because you cannot screen billions of sequences. And so that's and what we are developing is some sort of a way to, given a sequence, produce some score of this gene to actually function in food. And that's what we are doing at Flourish, and we have a few examples in which it actually works. But this computational infrastructure is actually taking care of many steps along the way that also require prediction. This is just an example of our methylcellulose protein that we have in-house. What you see here is rheology. On the x-axis, this is temperature. Y-axis is storage modulus, is like essentially what happens to the stiffness of the protein in very general. And once you heat it up, it becomes more firm. And the methylcellulose is the blue line. Our Shiru protein is in green. And the question is what happens when you cook something? You need it to start being stiff in a certain temperature, and that this needs to correspond with the requirements of whatever company that wants to replace methylcellulose with a protein product. And this is what we have found. One of the things that I want to put to dedicate 5 minutes to is one of these components along the path from sequence to function in food is protein localization, which is it's an old problem, quote unquote. But if I know where the sequence ends up in the cell, it can help guide how do we separate the protein from the biomass, and also help get rid of all these, for instance, membrane-associated protein that are very hard to work with. So we want to avoid them. So this helps choose the right sequences and get rid of the wrong ones. And that's one example of an application of Flourish. And this is a paper that we published back at 2022. The basic question is given a sequence, where will it end up in the cell? Traditional approaches, maybe some of you have had the chance to work on these. The localization of a protein is in many cases associated with a small segment in the protein, which is a localization signal, which is essentially some sort of a key card, which is part of the protein that takes care of shuttling the protein to the right compartment. And many of the traditional tools are based on sequence recognition, nuclear localization, nuclear exports, exports from the cell, like signal peptides. More modern approaches, very recent ones, actually turn this problem into a multi-class classification problem and use language models to represent the proteins as vectors and try, and using label data, predict where the protein will end up. So this is a work, this is like a database and a work called the DeepLock2, also a recent publication. And the thing is that the traditional approaches, they are successful, but sometimes they are not that successful because you can find proteins in the nucleus without a signal peptide. So how did they, without not signal peptide, nuclear localization signal. And how did it end up there? So the reason in many cases is that this protein interacted with another protein that had a localization signal, and then they went inside. So all these motif recognition based approaches will miss these. So this is where taking things like language models or structural information can help us dig out these cases. What we use is which was added to this language model, basic model for localization prediction, is actually taking structural information into account. So here you see how we turned protein AlphaFold model structures from protein into a graph. So based on an adjacency matrix, which is essentially all amino acids over all amino acids and the distances between all amino acids in the AlphaFold model. This can turn into, like, we keep only a couple based on a cutoff of the distance between the amino acids. And essentially, an adjacency matrix can be represented as a graph. So any protein for which we know the cellular localization, we have a graph. The edge feature is essentially a distance, or like is a binary sequence, whether it's close or remote. What we added upon existing similar solutions is node features for this graph that represent a protein that includes the dihedral angle, which is like the arrangements of the amino acid that can be inferred from the AlphaFold model, as well as the confidence, the PLDT score, which is like how confident AlphaFold is in the location of the amino acid. This corresponds with the disorder of the amino acid, and disorder, it can be associated to some extent, like many of the signals that help protein shuttle into different compartments are disordered. And also positional encoding, which is also very important. In adjacency matrix, you don't have the primary sequence. Two amino acids can be very close to one another and be very remote in terms of like the primary sequence. But many pairs will be very similar, very close, because they are just one after the other in the primary sequence. So you don't have it in the adjacency matrix. So we added also positional encoding. So this is, I will not expand a lot on this model, like the details are in the paper, but essentially we, in broad strokes, assembled both the protein language model that represents the protein and also the structural information to come up with a model that can be trained on 24,000 proteins with known localization and also tested. And eventually, like when you look at the results, we have tested all these, the prediction on a test set. So we were based on DeepLock, which is the biggest database of protein localization, and tested it on human proteins, which is a separate database that has like also localization signals. So this is data that the algorithm did not see before. And GVP alone is the structure-only based prediction, which has a F1 score, which is it's significant, but it is not as high as using only the language model baseline. This is the result of DeepLock 2, which is the algorithm that they used, and this is the architecture that ensembles both structural information and sequence information together, and we have like improved the F1 score. Yeah, so I'll finish here. Take-home messages are first, this architecture of ensembling different types of sequence and structural information and trying to do prediction based on labeled data that we are generating inside at Chiru is an architecture that is generalizable, and we can use it for other prediction tasks. Again, like we are feeding the different components of Flourish of prediction by different information that is the result of the experiments that we do on hundreds of proteins all the time that makes all the weights better of the prediction of different components along the way. And finally, as a bioinformatician, I didn't have a chance to eat a lot of the products that I was part of the team, and this time I'm even part of the tasting panel at Chiru, and I actually have like I can score like, oh, this is better, this is worse, and that's an awesome opportunity. And we have quite a few very promising ingredients that will end up being in these small last few words in ingredients list from Chiru. Yeah. Thank you.
That was great. If I understand correctly, the goal is to produce the proteins of interest recombinantly, and that would be the way they would be produced for food.
Yes.
Okay.
Yes.
And is there an example of any protein that is produced recombinantly at economic scale that is used in a foodstuff now?
Yeah, so one example could be soy leg hemoglobin produced by Impossible Foods. And that's what brings...
And they make that recombinantly, they don't extract it from peas?
Yeah.
Okay.
From soy.
Yeah.
Yeah. So that's part of the ingredients. And they are like...
Sorry,
I think...
I said economically. Right? I mean, famously, Impossible isn't doing it particularly efficiently.
Yes, there are definitely... All the field of protein fermentation is like... There are challenges that many companies are working on trying to tackle. And obviously, like, you want lower use rate. That's like so if a gelation agent needs to be 10% of the product, not viable, you need to go much lower and do the economics.
Yeah.
There are, I think, 5 proteins now that have GRAS approval produced by fermentation and are part of food.
Fantastic. Thank you for the answer.
Yeah.
Anything else? All right, thank you. All right, our next speaker is Trent Hauk, who will be talking from Where True Technologies, telling us about some interesting ideas of bringing bioinformatics tools into SQL.
Thank you. Thank you, buddy. Okay.
Cool.
Yeah, and now if you could stay on this
Yep.
side, this side of the line, I'll have a better shot. Thank you.
Yep. Awesome. Yep, thank you.
You guys ever examine it at the same time?
We were. Awesome. Yeah. So thanks everybody for being here. I won't spend a ton of time talking. I'll try to move through this fairly quickly, but definitely happy to talk to anybody after this. So yeah, just sort of brief introduction to me. My name's Trent. Run a company called Where True Technologies. Our basic ideas is what I'll get to in this talk. Previously, sort of software engineer, data science at a couple of biotechs, and then before then, worked on sort of like recommendation engines. So like you might also like this thing. Hopefully not more vacuums if you bought a vacuum, but vacuum bags if you bought a vacuum. Quickly, so Where True Technologies has 2 tools, bioinformatics tool and cheminformatics. So doing, say, like alignment or reading FASTA files in the bioinformatics tool, or doing, say, like Tanimoto similarity and reading SDF files for cheminformatics. And then we have a couple open source tools if you want to actually dive into code. I don't know how much people here care about code, but there it is. So.
Pressing the right arrow.
Pressing the right arrow.
Excellent. Press the bottom arrow. Try it now.
Okay, yes. Do that. Okay, sweet. Okay, so maybe I'll talk a little bit about sort of the state of things. Won't spend a ton of time here because I think actually, unsurprisingly, Peter did a great job of sort of talking about this. But just to sort of summarize, if we sort of break down maybe like the types of bioinformatics that takes place, we can maybe break it down into 3 categories where we have like primary analysis. So this is things like base calling, where you're just basically saying, can we generate like the very basic data that allows us to do the analysis? Secondary analysis, where we might actually try to do, say, like the annotations of on a genome to say, here are the proteins, or here are the HMM that hopefully dictate some of the function on the protein. Generally tends to be some computational step, tends to be very like sample based. So like you will run a sample, you will get the results. You might then use that to feed back into some additional experiment. You will then sort of do this cycle over and over again. And then we have maybe the idea of like tertiary analysis, where we're trying to say, okay, like we've generated a huge protein data set, or we've generated a bunch of, say, variants for human genomes, and we want to be able to combine those and generate sort of larger insights based on that data. And yeah, sort of successive complexity and successive size of the data. And I would argue, and part of my career has basically been built off of taking sort of primary and secondary and tertiary analysis and scaling it up with modern, people say modern when they want you to not think about the other tools and think their tool is better. So that's why I'll say say modern. But yeah, sort of bringing those in and using tools that are fairly ubiquitous in other industries and to bioinformatics. And so if you'll forgive me, I'll talk maybe quickly about data science and the sort of evolution of data science and analysis and how that's it's somewhat been a parallel track with respect to bioinformatics. So folks who have done data science, and some folks might use these tools now, but you have sort of pandas and Spark and dplyr and awk and sed and using sort of flat files, and those have worked relatively well, but at some level they've sort of ignored 50 or 60 years of database research for, say, like OLAP databases, online analytical processing databases, which is maybe different from like a transactional database. And so these are databases which can scan through large amounts of data very quickly. They don't operate on rows, they operate on chunks and columns to work work quickly. Join optimization, I'll talk a little bit about a problem where join optimization really matters, and can basically be the difference between your ability to have some analysis work locally and have that be then transferred to the data set that you would actually try to analyze. So sort of bigger than memory data sets, scalar function, out of memory, indexes, right? So like I'll show an example, or I'll have some example here in a minute, but bioinformaticians, well, let's just go to it, right? So you have like flat files plus index files, right? So you have a VCF file, you're generating an index file for that. You have a BAM file, you're generating an index for that, right? So there's sort of been these parallel tracks of software development where there's certainly been a lot of like great work and it's been super critical, but it has maybe not swerved into database software and database research. And again, it's sort of my view, and the reason why I started Where True is the thinking that there is an opportunity there to improve how people do data analysis and the ease of data analysis if we can sort of come to the light. Right, and so often what we find too is there's folks who, CLI ninjas, if you will. I'm sure some folks are that, and that's great. And maybe like I do now, I like type it into ChatGPT. I'm like, how do I create an index from this VCF? And that works really well. Or sort of distinct storage between like metadata, right? So like your ELN or your LIMS, which has like your sample metadata, and then you have your experimental data, which is say like a FASTA file, a GFF, an HMM, or whatever sort of the output of the particular analysis. And really right now, in a lot of cases, those live in like two different places, right? So the one of them lives in your Postgres database, your MySQL database, your Oracle database, or whatever. And the other one lives, say, on some file server or S3 if you're using a cloud service provider or whatnot. And so, yeah, there's been these like roughly parallel tracks in my view, and it's sort of my hope that we can use SQL to sort of bring that together. Okay, so kind of maybe I'll give some examples of kind of like where we're going or where I'm hoping we can go. And so, because I made this presentation, I can be a little bit slanted in how I present things. So, right, so if we look at like secondary analysis, which I mentioned, potentially do like homology-based search or like alignment score, right? So there's no reason why those types of scoring that you're doing can't be pushed into, say, scalar functions that are available in SQL, right? So you can imagine cases where, yeah, this is the sort of Panama banana is kind of like the classic alignment example, but you can imagine, say, running this on your table of primers, running this on your table of DNA sequences, rather than kind of pulling that out into a FASTA file, then doing the analysis, finding what you want, and sort of pushing that back into a downstream system. And so, yeah, the ability to sort of write SQL and do that computation in the same data set or the same same computer, if you will, because right, a database really is just like a program running on a machine, and what we do is more or less like augment those with code. So like homology search. Another example, and I would say this is not possible yet with our tools, but indexes, right? So like you have a genes table where so this is possible, but you can basically create a genes table as a create table statement. It would be excellent if you had a database index and you are using an index which you create on a particular column of your underlying sequence. And then based off that index, you can run searches on that, right? And so database indexes work really well. And like if you are performance tuning a traditional database and you have sets of queries based on the access patterns you're seeing, you can potentially add indices to improve performance. There's no real reason why, given what's possible, and again, these are just code. If you can edit the code and build these, you can do stuff like this. And so this is kind of the idea of secondary analysis, where we can potentially use BLAST, do the analysis in SQL, and sort of save a lot of sort of schlepping around various data sources. Right, so then you look at like tertiary analysis, right? And so this is sort of combining several ideas of secondary outputs, cross-sample, and trying to get some deeper analysis. I'll sort of give an example, and this is again where I can like choose what to show you, so it's too small to look at, but it's on purpose. But like I'll just sort of walk through the basic idea. This is actually very, very similar to a little bit what Peter talked about, where you have a config, right? You have some contigs, you have a GFF file, say based off PROCA, which are the gene predictions. You have a FASTA file with amino acids, you run HMMs on that, and then you're trying to more or less join all those data sets back together because, say, you want to find operons that might encode, so at Mammoth Bio, where I was at, say have like 2 or 3 Cas enzymes that you might care about, or say you're looking for natural products and you know that there are certain PFAMs that are indicative of a type of natural product. You can potentially use PFAM to say, okay, if they have these 3 PFAMs, it's likely that that's a good candidate for sort of some downstream selection. Just to sort of show one of these, and this is kind of what I talked about. I don't know if folks are familiar with like dictionaries or hash maps, and so these are very useful data structures because they can allow fast access, so you more or less create keys and values. And that's kind of how you would naively implement a join in some code. The problem with that is you're more or less loading all of that into memory, and so you get that speed. The problem is, even on like modern computers, so I have a fairly new MacBook that has 24 gigs of RAM, which is pretty good for a local machine. That doesn't really scale up very well, right? So if you start having fairly large data sets, this breaks down really quickly. So either you can't do this analysis locally, or even if you potentially do, say on a test set, how do you scale that up to larger data sets? And so this idea again of joining data back together can be sort of expressed in SQL, which is sort of this idea where you can read in a GFF table locally. You can then join that with, say, the output of the domains from an HMM output, so that the tab output. You then say can potentially join your locus tag with the target of the PFAM, grouped by the reference sequence name. So this would be the contig in this case. You can then read your contigs, do a join, and then say, okay, where are my contigs join on this PFAM table, where the list of contig PFAMs contains, say, 1 or more, and right, so then you can have a where clause, and you can potentially add multiple predicates here if you have an idea that some contig has 2, 3, or 4 PFAMs that you care about. And using underlying sort of databases, database tooling, this works locally because it can offload to memory, or excuse me, offload to disk in cases where with a hash map or a dictionary, you would sort of run out of space. And so this also, again, I get to sort of cherry pick a little bit giving the presentation, but it is sort of is quite a bit smaller. Is, in my opinion, SQL is maybe easier than Python. People like disagree to some extent, but this can be run in an editor, and again, can be run locally, but scale really well to much larger data sets. So I'm just gonna, so sort of quickly, again, trying to go fast here, but there are like a number of other advantages in my viewpoint as you sort of move to SQL. So there's obviously a number of off-the-shelf tools for visualization, right? So if you can, say, visualize a GFF just by putting it into, say, Tableau, that gives you a way to use, your off-the-shelf tool for visualization. If you write stuff in SQL, you can write it in really fast languages and then run that in, say, Python. So you get the speed advantages of Rust is kind of the hot new thing, but a fair amount of the code is also in C++. So you get fast code while being able to potentially use higher level languages to express, the glue. Again, lots of off-the-shelf tools for ETL exist. People, like I mentioned earlier, are already using metadata or SQL for metadata. Being language agnostic, and then fair data practices where the data is easy to find, it's easy to interpret, it's easy to track down, and so forth. So really, like, I know that was like super duper fast, but I just kind of wanted to run through a couple examples of my hope is that we can sort of take SQL and take bioinformatics and move it sort of up a layer, and hopefully in the process, basically make things easier. And that's the whirlwind.
I really liked your talk. I'm a huge database nerd. Okay. I like write SQL for fun and do like horrible with recursive queries for fun, and fall asleep to database lectures every night to the sheer terror of my own father. I'm curious about two things. You showed a lot of Python and other, like, it is my understanding that bioinformatics guys like R. What are your thoughts on technologies where you're allowed to embed script engines into databases themselves so that you're not, as you said before, schlepping around data? The one I'm thinking of specifically is GraalVM. And then do you have any comments about getting the data into the database and normalized across, from machines? How do you get that last mile working well? Because for me, that's a huge pain in the ass, because there's some dude with a USB drive running around that's part of your ETL pipeline.
So I guess I would say to the first, if it gets the job done, that is great. So being able to embed, say, your scripting language inside a database is good, and you are seeing, for example, with Postgres, which is maybe a little bit sort of more on the sort of forefront of these tools, the ability actually just recently to embed something like Rust as a sort of trusted language, and so that's pretty exciting to see. I think until that sort of matures and allows, I guess I should say a couple things. One, the intersection of people who can write Rust and are really good at biology is very, very small. I'm not great at biology. I come with a sort of a software background, and so that's really tough. And if you're not doing Rust, you might be doing Python or R, which again, if it gets the job done, absolutely fantastic. I'm all about sort of practicality. You are then obviously going to give up performance by using those languages and embedding them. So it's sort of, I would say, great for one-offs. It's sort of my opinion, if you really care and you're going to do it a lot, that's maybe where you want to just go ahead and say okay, we're going to express it in SQL, or sort of express it in the underlying language and then allow SQL to be written on top of it. It's also, and I might be a little bit out of date on this, but the ability to sort of so like there's scripts which you're writing, which maybe you can say version control and have like good code practices around. There's sort of SQL, which sometimes exists outside of that. And there's sort of some like kind of what you're describing, which is maybe some like weird middle ground between the two. And so having sort of good coding practices around that is maybe kind of challenging. Yeah, to your other question around schlepping data around and normalization, it's a really interesting one. I think in a lot of cases you can use SQL as the sort of like normalization construct where you then are sort of say bringing some data in, joining that as part of your ETL pipeline. That is sort of how you're normalizing, and then you're pushing those data to like different tables in a normalized fashion. The machine question is super interesting. I don't have a great answer for like actual like machine. I mean, there are certainly like products coming out of say like AWS that try to help facilitate that sort of on-prem to cloud. And if you can say get it to S3, then these tools can work really well. But the sort of like person who is in the lab setting up some agent on the sequencer, is, I guess, outside of the scope, if you will, of the problems I'm trying to solve. Not to say it's not important and not a problem, but yeah, I don't want to be that person, so I try to, so I'm not solving that problem.
Hi, so I'm a novice coder myself, and lately it's been really nice having a tool like ChatGPT to be able to help fill in some of the blanks. Yep. So I'm not banging my head on the keyboard for about 8 hours. But for SQL, is that something that GPT can be used for, or Copilot, or do you trust that it can like be able to pull or summarize some of the data sets that you have in order to get the data that's correct? Or do they have hallucinations just as much as the others?
So I would say generally, it's one of those things where you should be highly skeptical, but I do think it generally does do a good job. So like if you can ask it to generate some SQL to be like, I want to say, take this data set and join it with that data set, it can generally do the right thing. You should have mechanisms by which you can QC the results you get to make sure it's doing the right thing. But no, absolutely, I think it does do a good job. There are even quite a few startups right now trying to do sort of like text to SQL, if you will, where you can sort of express your question in prose or sort of human language, and it will break that down into SQL. I think those are interesting. Part of the challenge with something like this is it's sort of adding in new functions, both sort of new table functions and new scalar functions, which aren't part of the general SQL standard, but definitely possible. I would be lying if I didn't say Copilot wrote a fair amount of like the underlying code in this, because it is a great time saver and does generally good work.
Okay, one more. Yeah.
Just curious if you had any speed benchmarks on your system compared to a non-SQL system?
So say it's, depends on like the tooling of course, but say like if you are trying to maybe parse through like a FASTA file with BioPython, which is sort of like the Python common one, we're about like 5 to 6 to 7 times faster than that. Sort of say like your sort of SAM tools view, we're sort of on par roughly with that. So it's not, those are written in C, so they're quite fast, and they've been around for a while, so they have like a fair amount of optimization built in. So I can't really say we're going to have a faster tool than that. I think where I try to maybe skirt around that question slightly and say like, once you're trying to do something beyond just like SAM tools view and say trying to join that output with something else, that's where both like the developer time and the speed time start to sort of, in my opinion, weigh more towards this. So again, tools that are already in C, it's about on par. Tools that are say in R or Python, generally a pretty good speed up.
All right, thank you, Trent.
Yep.
All right, so now we'll have our panel. So we're going to have an interesting discussion on longevity industry and funding and VCs and things like that. So if the panelists want to come up here.
Now, if everyone wants to take a quick break, if you need bathrooms, they're around this hexagons here at any of the orange frame doors, and also down the hallway. Just take a quick one, and then we'll jump in.
And then, should we put this? Yeah.
It's got a really good program.
This is great.
Super interesting.
Great job on the AV.
Yeah, thank you.
It really looks good. I can't wait to see the
Sounds
video.
good too.
I have a picture of our panel when we're here. Where it's like more recorded and it's going to be...
You can take a picture, you can, whatever you want to do.
If I may just ask you to take one picture of us when we'll be over here.
Oh, you want
or
me to take a
screenshot,
picture?
Or
Oh, yes, I
while
can.
I'll be over here.
On my phone, and then I'll send it to the group?
Like that. Thank you.
Perfect, yes.
Thank you so much.
Very good. I'm glad you thought of that.
Yeah, thank
I don't
you.
always. So the panelists will just pass this microphone as you speak.
Okay.
Just keep it, close-ish to yourself. And then this is for you, Shane. Yeah. Whenever. I'm sorry to interrupt you. Yeah. Are you on the panel? Wonderful. We're just here for the drinks. For the snacks. Very good. Well, when you speak, just make sure to bring the mic over to yourself. And
Do we have to move the mic every time somebody starts talking?
You don't have to, but if you want to be heard on the stream, it would
Well,
be helpful.
I am thinking if someone might
Like,
forget.
it can't just be placed in a central location and hear us all equally.
If you want to
The
move
reason that I
your
asked, Phil, is that there's, okay, like, I understand your means of production concerns about the sound quality of the outcome,
I want to appropriate the means of
right?
production.
And my concerns about the spontaneity and, like, normalcy of conversation and people not handing the talking stick around.
Well, we'll land somewhere in the middle. I suspect.
Okay.
Yeah. If someone's talking a long time, just
Sounds good.
If there's a lot of rapid fire, then it's cool.
Yeah.
Fair enough. That's the thing that I was concerned about. It's like,
I like how you navigated that.
my concern is if they have to move, someone will forget. Like for sure, if they are really enjoying the conversation, they will forget to move it. So, like if you want people to move, you should have this.
Yeah.
Like,
What we're gonna do is put it really far from you, Chris.
that way I can say anything I want.
You could say, no one's gonna hear you. Okay.
They'll hear me. But thank you very much for the explanation. I think that's going to be fine. Can I have a bottle of water?
My pleasure. We're gonna make sure that you sound
Well, do we have just the three of us, or is there a fourth?
Shane's gonna moderate.
Oh, okay.
Yeah. And he has his own mic, so he can bum rush. He can shout you down. I mean, the sort of acoustic epicenter here will be like this.
Yeah, if we're not doing crossfire, I think it'll be fine to move around.
It's all gonna work out. It's all gonna
Does
work
it
out.
slide? It doesn't make sound when it's. Should we lift it? Should we
Just lift it.
lift and drop?
lift
Okay,
and
not
drop.
slide.
Yeah, I wouldn't slide it.
Okay.
But don't let this distract from the content. It's all content driven.
Guys from Launch, I just wanted to ask, is anyone going to the Bay Area aging meeting tomorrow? Yes? Yeah, I was just trying to get like a schedule of that day. Is that, can I get it somewhere? Because it's like, I'm gonna need to take a bus there, and it takes like 2.5 hours to get there.
Well, good luck.
Yeah, exactly. So I'm just gonna have to like get up really early, and I was just like, could I just miss something at the beginning?
There's probably an email when you register. Did they not include a schedule? Or?
Yeah.
Well, what's her name? What's her name? Sent around email like recently.
Yeah, I have that one. Do you want to enter your email? I will just.
Yes.
Just send it to yourself. Forward it to yourself.
But the short version is it starts at 9.
All right.
You can just forward the email.
Is it here like that?
Let me help. Sorry.
Yeah.
Yeah.
I have
I'll do that. Let me just.
So I'm just visiting here from Czech Republic. So it's just like.
You're from Czech Republic?
Yes.
Oh, nice. My brother lives in Prague.
Wow.
He finished this higher school of economics.
Oh, cool.
free to, riff off each other or whatever.
Yeah, I study and live in Prague as well.
I don't usually prepare.
I'm just here for a couple of weeks for the On Deck Founders.
Oh, nice. I've been on Deck Longevity Biotech while they had it.
Yes. I know Nathan Chang.
Listening to Lex Friedman's interview with Steve Wolfram.
Oh yeah.
Do you listen to the Lex Friedman show at all? Podcast?
No, I see it probably come up, but I haven't.
Yeah, my brother actually works in
really
our
nice
company as well.
I think it was like 4 hours
and we have
long.
also a second guy from Prague. So we have like a small office in Prague actually. Two guys are working from there.
Oh, that's awesome. Like I would like to set up something in
modeling actually.
like some office as well, but it's just me currently and one guy who's at the university, so he works there his PhD. So
I like.
He can write to me if you want an introduction.
I mean, he's really pushing on like the.
Oh, okay. I will. So I have your email now.
Yep.
Perfect.
Good.
Excited for the talk.
Sure.
But they were talking a lot about how you can actually hybridize them and work these them together. And I think that's like super important for modeling biology. Is that like you have all these different layers of abstraction, different aspects, kinetic model. So many different aspects. There's not one model that just can do it all. I'm curious to see whether you can take a large language model and use that as like a glue to like pull all of these different types of modeling together into one super model.
Yeah, I think that would be very powerful.
On our end, we believe that this is kind of like too much, and we try to do a much more simplistic approach. The reduction is a total reductionist approach. We believe that the strongest signals will be found first using this approach. It's like, so
we should save this debate for live.
Our scientific founder used to say that like if humans who invented a steam engine would know that there are molecules, they will never do that. They would just make a transphometer to measure each molecule velocity and speed, and try to figure it out how to derive all of the power from those molecules. So we believe that in biology, tools came first, and people just got caught in a trap of trying to measure as much stuff as they can, and now they can measure lots of stuff, and it's actually confusing. So we kind of try to just to turn it bottom up and identify things that are kept on both macroscopic and microscopic level. So not trying to extrapolate the results of experiments that are a week or 2 weeks long in a cell or in a model organism that doesn't leave the age of human, but rather flip it on the other side. Take the human data, look into there, and then go into basic model just to validate yourself, but not to discover something there. Yeah. So this is our approach, really, just from the other side.
Yeah. So I'll bring this question up. I think it's like some good conversation. Yeah.
How about you, Chris? What's BioAge's approach?
I didn't hear the whole.
Yeah, we've been discussing more like
different approaches to modeling, basically.
Like from to measure as much as you can, and then try to understand, or rather to measure all you can, but simplify it significantly before you try to understand how it works.
Well, we certainly measure all we can. How much do we simplify? I don't know how to answer that question. I mean,
that's kind of what a model does in a sense, right? Like,
yes. I mean, I guess like we're more interested in. Like we're asking what, so take 10,000 people and like take their health history and then take all of their molecular profiling over 50 years, measure all of that, ask what are the molecular differences at age 50 that predict whether or not you're going to make it to 70, and in what state of health you will be when you do. So that's what we do. I'm not sure how to answer your question, though.
The models that you guys use, I guess, are they?
I I mean, they're predictive. It's
There's
simple.
just
It's predictive.
statistical
It's predictive
association.
model. It's like the independent variables are all of the different molecular expression and metabolomics data, and the dependent variable is health outcome, right? So essentially, I mean, it's not a big linear regression, but sort of intellectually, it's a lot like that.
You're not actually trying to like build like a metabolomic
No,
pathway
no, fuck no.
thing, network science type thing. No,
So
just
simplistic.
statistical.
So kind of
statistical.
simplistic then, in
It's like
that direction.
like we make no. It's weird. Like we make no model of how the system is working. Like there's no entity in our modeling that is like a simulated, even a very abstracted simulation of what's happening.
Yeah, it's just math.
It's just it's just arithmetic.
How you guys doing? You guys ready?
Sure.
Can I get you some more waters, or I can go chew
You guys are recording, right? We started 5 minutes.
We'll use Mid-Journey to put the audience into the seats. I heard a new version just came out today.
Yeah, it's exciting. Yeah, they gotten better with the hands now.
The hands are the greatest. The teeth are also a giveaway. If you look at a beautiful woman or whatever, like all of her teeth will be exactly the same size and shape. But it does amazing with faces otherwise, I think. Midjourney has been doing really well. Especially if you apply something that's like a filter, so it looks a little bit unnatural to begin with, and then it's like indistinguishable from a real picture, except for the teeth and the hands. And they have too many teeth, also.
Yeah, that's creepy something.
It's all, the uncanny valley is real.
From Stanford.
Do you know this concept of the uncanny valley?
Red shirt.
No, the uncanny valley. So the idea, it's basically a graph. So the
That was an
graph
interesting
here
talk.
is
I felt like
how
connected the history.
similar to a human face. Okay, this is perfect identity to human face. Is this y-axis, and then further and further away, you're more and more distant from a human face. So over here, it's just like a circle with 2 dots and a smile, right? It doesn't look anything like a human face. This is not
Just,
unsettling
taking
to
the
people,
data,
all right?
it's
A
one
human
directional
face, not unsettling
process,
to
and then
people.
ending
It
with
turns
the
out there's a
looking
point
at it.
in the curve where like
That's
you
what I was
get
trying to get at with my
close to a
question,
human
like
face,
iteration.
people get very bothered by it. Like that's really.
Gets to 99%.
So like
They try to make it not too human, right?
yeah, exactly. So if you can't be perfectly human, you need to give a good deal of room, so you're not like creepy.
I'm not like up to date with the field.
And somebody made that's the uncanny valley. You're first to dip in this graph is the valley.
Yeah, that's pretty much
And then
where it's
somebody
at
made it
right
interesting.
now.
It was a joke point, but
You
it was
touched on
very
the idea
funny.
of the automated modeling.
Yeah.
So why
That
is
is
this?
an area that I
Why
think
is this
is exciting.
phenomenon? Why is it true?
People are not only
that
building the models
faces in the
automatically,
uncanny
but they
valley
are
are
starting to
disturbing.
incorporate other omics
And
data
somebody said, well,
into that model
it
to
means
refine
that at
the
some
model.
point in human evolution,
that cycle
it was
where the
important
model
to
would
distinguish between a human
actually
being
flag
and something that
things
looked a
that were
lot like
you
a
should
human
test
being, but
this.
wasn't.
Oh yeah.
Right, like it's like I don't know if I'm right. Test it.
So
in
like
reality.
competitive species, like
That's what we did
or
at Simple.
other
We were,
monkeys that
in
look
a very
like us.
simple case.
Yeah, monkeys don't have facial recognition, so it would have to be like hominins. But like, I think the joke was intended to point to things like a vampire. But yeah, sure. Like, we'd want to know. You'd want to be sensitively. You want to be very sensitive to like, is that a human or is it like?
Is it my
the
species?
dude from the next town over? Yeah.
Every run. And just like
Interesting. I heard that people got pretty
like, oh, you can actually lower this region by 50% and it has no effect.
I had not realized this was going to be this
A
long.
slight change in temperature like reduces this byproduct and things like that. It was all fully an automated loop where like it was like generating sort of its own perturbations and then trying to like make its own.
Just not my,
That's the kind of we should be doing
Yeah.
that for biology.
If I weren't afraid we're on a hot mic, I'd say some things about it. Yeah, it's not my topic.
Yeah, no, you're totally right. So you could imagine annotation as a self-driving lab type approach. Yeah. I don't think anybody's really done that.
I heard that Calico is trying to make like a computational model of the C. elegans, but I don't know how. I just heard that they're working on it.
Yeah. Well, there's also a group at Google X that's doing cell models.
Well, maybe that's the same. It might be
I think they're different.
Different? Two different? Okay.
Different campuses.
Well, I mean, sometimes that's good, actually. 'Cause
Yeah. So, bringing in the omics data with the machine learning to improve the models is a good way then to drive better modeling, better imitation. You could also imagine it's a lot of these missing genes and things are probably because it wasn't done with long-read sequencing or something.
Yeah, so
and
the-
so that's
the assembly is broken.
Yeah, you can imagine having that as another way to refine.
It's crazy to me that those are still problems. Like short read, like sequencing is still a problem. Did they have like the sort of paired end things and all this stuff to try to solve these sequence quality problems?
Yeah, so paired end improved a little bit, and nowadays they use
Great. I was just on the phone with her today. We were just catching up.
They started doing that.
Yeah, her
and then
company,
they found
her
out that the
consultancy is
long-read
doing really
itself
well.
could be even better.
Nice.
And
So
now
yeah,
you
she's
can
really
get what are
happy.
called kilometers to
I'm glad that that
assemblies
referral worked out for you guys.
Yeah, I did. I like.
So
She
it's
was
vastly
exactly the
improved,
person I was
and it's
looking for.
probably nearing
Who's like,
something like
can
perfection.
just help me, you know, just
but
use the right words.
many
I
of
have
these
the
old
ideas,
genomes
but
that
I
he's
don't
still
have
working with
the
are not
domain.
made
They weren't
sure.
done that way.
Sure.
So I
No,
love
she's
it.
amazing.
So just
Well, I'm very glad to
rerunning
hear
the sequencing,
Very glad to hear it.
feeding that back through could like maybe
You guys
they're,
got
like,
in at the early
some of their
stages when
numbers
she
are
was
just
still,
capped,
and she's starting to be very picky about it. So like her big client is in Citro.
Yeah.
So like
I think it would improve a
she's
lot.
working with some pretty
How much
established
is a
people, but she's
long-range
gonna save some
Well, I
room
guess
for like
Prokaryote
small
is
startups like you guys because she's excited by
Prokaryote is
you know
very
the thing
cheap,
that
yeah,
isn't
but
what
to do it
everyone
on
else is
bigger
doing.
organizations, it's so
Basically,
expensive.
Yeah.
This barstool is gonna kill my head.
Is it Oxford and PacBio are the still
I might
the
have to
two?
stand.
I really like.
No, it's okay. Like
I remember
barstools
when,
are just like my hip. I
2008
have to
when
have my
there
hip
was, like,
replaced.
30
And
sequence
barstools.
startups all, like,
No, I'm gonna have it done
You're
next
gonna
month.
have
But
it,
the
yeah.
injury makes this kind of
Yeah,
very
there is a few
challenging.
new ones. There's Ultima and Element Biosciences.
I might. We'll see.
What's the what? There was one with like the spinning disc.
If I sit
thing.
further back
Nobody
on
will
it,
blame you for it. So
it's
there
not
was
just
a new
chemistry.
Oh yeah, I remember that. I
I
remember
just-
that. Yeah.
I just remember the spinning disc. I don't remember why or what it's
Yeah. I think we should. Yeah. Where's Pat? Oh yeah. Let me just.
Ouch.
All right, ready, everybody?
I need this.
All right, everyone, thank you. So we have our longevity panel here. So first of all, thank you, gentlemen, for coming and speaking with us. I think maybe we just started out by having each of you go and if you could introduce yourself a little bit and where you're from, and maybe talk a little bit about just what got you into longevity and what are some things that are really exciting in your current role. I think we're going to pass that around.
Sure.
Okay.
Can
All right,
you
well,
do the introductions then?
Yeah. Well, I'll include a comment on that in my introduction. So my name's Mark Hämäläinen. I got interested in longevity just from reading science fiction and realizing that I was going to be dead before all these cool things were going to happen. And I thought, so rather than working on like space technology, I should work on longevity first, so that I could actually live long enough to see things happen. And I started off by working in like a biology lab, but quickly realized that like the rate of progress was like way too slow, and ended up dropping out of a PhD and coming to California to work on technology development to try to make research go faster. And then another bottleneck though is just the number of people working on it. So I took a break from technology development, and now I'm helping people get in the field. So for people that are interested in longevity, I am co-director of an organization called the Longevity Biotech Fellowship, and we basically help people from other fields, whether it be software, chemistry, whatever you do, synthetic biology. We want to, and you're interested in longevity, we can sort of get you up to speed on the industry and how you might be able to contribute in a highly impactful way. And what is longevity? I don't know, maybe that's a semantic argument. How long do you live? How long do you want to live?
Hi everyone, my name is Alex. I'm Chief Business Officer at Gero, and Gero is a physics-enabled AI drug discovery company. We try to understand what aging is and what it is not and how it relates to diseases, and we want to develop drugs to slow down human aging but first to end a couple of diseases of aging. Last year we've done a deal with Pfizer. We just have raised Series A, and I just have moved over here to the Bay Area. I've been within the industry for the last 5 years. I actually I wanted to find something meaningful to work on, and I found it to be really motivating personally. So I just switched from my previous role, which was like more like in a production business, and I started my career as a management consultant in PwC, and in biotech, I'm like an alien in biotech, but it helps a lot, actually, I have to say. So at Gero, we believe that aging cannot be reversed. We believe that aging can be slowed down, and we're working on that mission. So to your question, what is longevity? It's just that, let's say, it's not an industry per se. Right now, it's more like a already big and sometimes well-funded enough group of people who look at aging as a problem, see it as a problem, and at least want to do something about it. So I would describe ourselves as this at this point.
Hi, everyone. I'm Chris Patel. I'm the Vice President of Media at BioAge. BioAge is a Series C, still privately held company, whose mission is to discover fundamental mechanisms of aging, design drugs to target those mechanisms, and then use those drugs to treat disease, and ultimately develop those drugs into something that can be used to slow or prevent many aspects of the aging process. I am a scientist by training, so I wasn't always the comms guy. I think that BioAge leadership made a decision around the time I was hired that it would be easier to take an expert in the biology of aging and train that person in media than it would to take an expert in media and train that person up on the biology of aging. And so far, it seems to be going pretty well. I have always been interested in aging since I became a biologist. I identified it as what I thought was the most interesting problem in biology. And at the time, which was a long time ago, before I'm looking around, before several of you were born, in the 80s, it was very understudied. And the sort of foundational observations of the early 90s that revealed that aging is something that can be subject to genetic analysis in the same way that oncology can be, hadn't yet been made. So I'm going to define the longevity biotech field as the set of companies that are operating according to one deep bet. And the deep bet is that aging is the premise is that aging is the primary risk factor for most diseases, most chronic diseases. And the bet is that those many diseases, from Alzheimer's to diabetes to kidney failure to you name it, that those myriad diseases are driven by a much smaller number of fundamental mechanisms. And that if we can target those mechanisms with drugs, we can treat many chronic diseases at once. And we can, now to define longevity in an important operational way, we can extend the health span of individuals, okay, which is the number of years you're both alive and free from chronic disease. We also wish to extend the lifespan. Turns out every way we know to do health span extension in experimental animals also extends lifespan. And so that's the paradigm that we're operating from.
You know, that was really interesting. You mentioned that when some of you got into this, it was very understudied. And it seems like lately there is a renaissance of longevity research and companies starting. And I'm wondering if you could maybe talk a little bit about, you know, do you agree with that? Why is that happening? And what are the key drivers of that?
Sure, I can start. I guess I'd say aging is probably the most complicated problem, or at least perceived to be one of the most complicated problems humans have ever tried to do something about. And so we had to like build up towards get that being a feasible, tractable problem. And so there's been a lot of cool technology development in omics and the understanding of biology and the building of models of biology. I think about like measurement, and then models, and then modalities. So it's like we have like gene therapy is like a new modality. So there's all these like technology developments in measurement models and modalities that have improved. And now I think it's seen as tractable. And so I think that's why in the past it would be hard to get, like to say you wanted to extend longevity, people would just kind of laugh at you or not take you seriously. And most of the people that were doing it, actually, you probably shouldn't have taken seriously. But that has changed. I think we've gotten to the point where we could actually make substantial progress in the coming years and decades. So it's exciting, and energy, money, people are flowing in now because of that. Any other?
It's pretty, yeah, I do agree with Mark.
Okay. Could I ask you, what do you think are some of the key breakthroughs then that are going on right now, or that maybe we're about to see? And what are some of the companies that are working on these things? And do they tend to be small startups, or do you think you're going to see these breakthroughs come from big companies?
When we rotate the starting position, like a poker game. You go ahead, Alex.
Okay. So I'm a bit biased in that because our company has like really differentiated view of aging, and we kind of a bit contrarian to the whole industry. So our take is that the major promise of the industry that aging will be reversed, and like 80 years old people will go running 20 years old again. Like this is the biggest promise of companies like Altus Lobster. We do not believe that is feasible. We believe that late life phenotype can be reversed, which is associated with all of those that Chris told, like diabetes and etc., kidney failure, those like bunch of diseases come all at the same time at the late life. And we believe that can be reversed, but what leads to the increase of that risk cannot be reversed, but can be slowed down. So this is kind of our take. We published a bunch of papers in Science and Nature Communications. We kind of built this take on aging, and we will see if we are right or wrong, like within 5 years from now. So we are on the way to do that. And again, what was the second?
Yeah, what are some of the big breakthroughs, and whether they're large? Are you betting more on large or small companies?
Yeah, so from my perspective, but again, I'm totally biased, but from the inside, from my perspective, I believe we get a substantial chance that we getting it right. If we're getting it right, then our company will do that. so but I'm very biased, like I'm 100% sponsored by my company.
Yeah, I'm also paid to be an advocate for a company, so that's important to always keep in mind. Remember who's paying the check and whoever's speaking. But I think that the key breakthroughs really are, and this kind of gets toward the previous question. We have this convergence of technologies that allow measurements we couldn't do 5 years ago, with 30-ish years of fundamental biology of aging that went from like very, very foundational work to like very applied work, where we're actually extending the lifespans of animal models by 20-30%. Mouse, I mean, you can do a worm, I think 4 to 10 times, but then you're still a worm. And so, but I think that one of the things that's been frustrating to biologists of aging is that you have these interventions that you know can extend lifespan in a mouse, and it's like, well, where's the translation to human beings? And I think that one of the things that's been really powerful for a lot of companies is the realization that we have so much human data that we can, from biobanks that have been collected over 50 years, and we can look at that data and we can parse it and slice it in a million different ways that we couldn't very recently. And then you can start to ask questions like, okay, here are 10,000 people, and like at age 50, they're different, okay, but they're all reasonably healthy. What are the molecular differences in those people that predict whether some of them, make it to 70, like limping across the finish line, sick, and pretty soon after that dead, and the others make it into their 10th decade in reasonable good health, right? And if you can identify those differences, then you can ask, well, how can I make everyone more like those people, right? And then then you can ask, well, what modifications could I do in terms of like a pharmaceutical? What intervention could I do? And then you're on your way to finding a drug target. And then all you have to do is figure out what disease you want to treat to get past the regulators, right? And so, but the breakthroughs are really the application, I think, of data science.
Right.
Work in the crowd here. Data science to these giant data sets that we've collected, and really beginning to translate the biology of aging from the laboratory animal into the human.
Right.
I don't work for any particular company. I work for a nonprofit, so I'm going to give a contrarian answer that it's not startups or companies necessarily that are going to be the most important thing. I think they are very important, but also a lot of technology development comes from government-funded research, public databases that are not walled gardens or that are free to access. Like the Human Genome Project was a good example of that. I wish we had more of that, more big public databases that everybody had access to, not just like walled off inside of one company or another.
I want to be contrarian about your contrarianism, if that's okay. I think Mark's point is very well taken that, I think this group, you use GitHub, you use all these public repositories, that the more eyes on something, the better it is. And the more people have access to data, the more the data can sing. It is true that some companies are proprietary about their data, but it's also true that within longevity, as a field, there's a growing effort to create common data sets and common reference points and common sets of biomarkers and common sets of clinical protocols so that when someone does a clinical trial, it's easy to compare it to a clinical trial that's done by somebody else, and that these studies that are done of people that are longitudinal can be compared. So I very much agree that the public sector has a huge thing to contribute. I mean, none of us, by the way, everyone knows the public sector is a giant risk subsidy for biotech. Like biotech, as we know it, could not exist without the public sector. Absolutely, right? Like libertarians can, well, I like to have a word with those people, And, but, I think that it is not true that every company is a walled garden. That's all I wanted to really say.
Thinking about some of the things that are driving the advances right now, we were having a good conversation earlier about modeling and what's been happening with language models and things like that. And I was wondering if you could talk a little bit about that and how do you see these things in AI impacting the longevity field, and what direction do you think that could take us?
Whose turn to go first?
You're good. You got AI in your domain name.
Yeah, that is right. So, okay. And again, we are taking a contrarian position so AI for drug discovery, hype word, right? We have several types of companies. Two major types are small molecule or antibody discovery companies, so molecule discovery enhanced by machine learning. And another big bunch of companies are target discovery companies, so let's understand biology better of that. So again, we can do pretty decent small molecule drug discovery. We've done lots of that, but we do not believe that there will be created a big monopoly in specific molecule discovery, because the chemical space is so vast that we believe every company which will attempt will carve out its own piece of chemical space and will be able to identify molecules in there. We believe that target discovery is the holy grail of AI drug discovery. We believe that it will create the major value for the industry, because we don't deal with Pfizer. We are not the guys who will teach Pfizer how to do molecules. This is insane. Like, they kind of know how to do molecules. What they are actually lacking is this machine learning capabilities on human data and the new ways to interpret that data to drive the biological insights. So we believe that this is like the biggest thing that will happen, and AI for drug discovery holds a huge promise. As of now, nobody knows which company will actually solve it for the biggest problem, for the target discovery. There are companies like BioAge, like Gero, like Incitro, who are doubling down on that, and I believe this is the right direction of modeling. And again, just modeling human health in a way that can causally, as Chris told, can causally predict which genetic factors causally associated with certain disease progression is the way to go.
That's a good answer.
I'll hop into it. The holy grail being target discovery. I think you can think of it, I think of it as sequential generations of holy grails, and that might be the one that's the closest. I'm actually personally more excited about something Peter mentioned, which is computational models of biology. Like if we, and improvements in new modalities. So this is like looking at like next generation. It's not going to be in the next 5 years, but maybe it's in 10 or 20 years where we're thinking about building computational models of biology, because right now, if you're trying to do a lifespan study and you have to wait for a mouse to get old, and then, you know, it doesn't even apply to a human, like this is a very slow iteration time. And like, we're not going to make potentially a huge amount of progress in a normal human lifespan if we're relying on those types of studies. So I think the computational models are really important, but not just, I know you guys are, and then the new modalities, which would be things like gene therapy. So instead of interacting with biology on a chemical level, interacting on an information level, and learning how to like have like a model where you could like say like, oh, if we make this genetic modification, we know what it would do. And we're a long way from that, like totally admit it. But I'm curious if there's anybody here, come talk to me afterwards if you're interested in that stuff, because I want to connect people interested in that with projects.
I want to kind of argue with you on that, because to actually predict targets that will work, this is what you need. You need to model human health in that way, just as you describe it. So I believe we are not like in conflict in here. So actually what we try to do, we try to create that model of human health that can answer what if questions. And this is the thing.
Yeah.
My contribution to this raging debate is that I think it is possible to make predictions without creating an explicit model of something. I think you can just do what essentially amounts to arithmetic. But what I wanted to actually go back to the original question about the role of AI in this space, and just address something that came up in a podcast interview that I did. Oh, by the way, as part of my responsibilities at BioAge, I host a podcast called Translating Aging, which is about essentially this. It's about the researchers, entrepreneurs, investors, even founders who are advancing the cause of longevity biotech in some way. And actually, both of the two people to my immediate left have been on the show at different times. Your boss,
my boss,
and Mark in his capacity as the founder of Less Death. This came up in my conversation with Jacob Kimmel from New Limit. He's the head of research there, and they were talking about the way that they want to kind of use generative AI to get out of doing experiments. And I think that's a little bit unfair of a characterization. So I'm making a straw man here. I want it to be clear that I'm not speaking on behalf of this person, but I'm riffing on something that came up in a conversation. When we think about how generative AI works, all right, there's always a gigantic training set relative to the thing the generative AI is doing for you, okay? So this is just kind of a caution to all of you bioinformaticians and aspiring data scientists. If somebody in longevity, or really any kind of biotech drug discovery, says, this biological problem is astronomically large, all right? We're going to address it by getting an AI to make predictions about the outcomes of experiments we've never performed based on the extant corpus of data that has been produced by similar experiments. It'd be like saying, all right, I trained my generative AI language model on one issue of the New York Times, and then I asked it to write Wikipedia, all right? The training set needs to be large relative to the output. I'm pretty sure I'm right about this. And there are people in the field, not the person that I just referred to, I'm again straw manning an aspect of what came up in that conversation, who are uncareful about this. So just have your skeptical hat on if you hear folks talking about that. But like, I think that, you know, as the corpus of data grows, we can use it to train generative AIs to do all manner of exciting things. And I think that's a very exciting direction. I think that, you know, as long as you're honest about, you know, Don't overfit and don't abuse your model, and you should be able to get some good stuff out of it.
One more comment on this. I agree with pretty much everything you just said, and I think it actually highlights the importance of that there's different types of models, and so generative AI is like super exciting, and everybody's like, obviously ChatGPT-4 is pretty like magical in certain ways, so that the results that it can put. But there's also symbolic methods, and I was just listening to like a podcast by Stephen Wolfram, interviewed by Lex Friedman, that came out recently that everybody should listen to. It's a really, really good podcast. But like you can hybridize different types of models, and I think like these large language models might be a way of like gluing together different types of models, where the large language model or the generative AI is actually making use of like kinetic models, symbolic models of the type of data that you guys produce, like, and it's all being sort of glued together. So yeah, generative AI by itself definitely has some limitations, and has definitely been hyped a lot lately. But it's also doing some pretty cool stuff. So like, I don't want to downplay it either.
I love it.
Yeah.
Thanks. I'd like to switch to the business side now for a little bit for one more question, then we'll try to take some questions from the audience. What kind of advice would you have for someone wanting to get into the field or maybe wanting to start a company? And what do you think about the current issues with the funding climate? Is that something that could be actually a danger to the field, or will this be a blip on the radar?
Yeah, so let me take it. To the first question, I believe the nice point to start as Mark's and Nathan's LBF nonprofit association. So it's basically a place where you can hang out with folks who are also interested in the thing and who run tons of companies in the space. So that is a nice place to start. And to the second one, I can share my opinion within the last half of a year. I've been talking to lots of venture capitalists in the field, and by the field I mean there are two groups of them. One group are ex-IT guys who went into longevity with a dream, and another group is life sciences biotech funds that just do their own thing and starting to look into that field as it emerges as well. So the risk profiles are completely different, and for different companies there will be different paths through both of these groups of people. But I do not think that for small companies current climate is going to make any change in that sense. It should bring more sanity into the industry for sure, but this is just for good.
I largely concur, and I think the squeeze is real. It is not an easy time to raise giant buckets of money right now. I think Alex is right that if you're not trying to raise that much money, there you can find, what did Barnum say about some type of person being born every minute? I'm just kidding. There's good reasons to fund at the seed level when you're not prepared to make a really large investment at the Series B, level. But when you get into the larger denominations, I think it's creating some friction. The environment is creating some friction right now. And that's why you really want to have, this is not the question of how to get into the business, but it's a question of how to survive. If you are an established entity in this space, you want to have a great team that understands not one thing, not just the IT thing, not just the biology thing, not just the business thing, not just the communications thing, obviously the most important thing, but still part of a team. And you want a team that is expert at all of these things, because there are a great many storms that need to be weathered.
Yeah, that's true.
And I'm confident in my own organization's ability to do that. Our peer organizations are doing a great job and doing great work. But I hope that things do turn around, because I would hate to see a promising company run out of runway. I don't know of a single company that's in danger of that happening, but I feel like if things go the way that they are, when you get to the point of companies needing, because when you're running multiple clinical trials, you need $100 million. It's not like you can go to the, First Republic, oh, just kidding, you can't go to them at all, and be like, I want $2 million as bridge. These are serious amounts of money, and I think I'm worried about that a little bit, but not existentially worried for myself, just more worried about the field.
thanks for plugging, so I didn't have to plug myself. But yeah, Nathan and I do run the Longevity Biotech Fellowship, and we've actually seen that a lot of companies are not hiring right now. Like, and one of our sort of processes has been to like help people find jobs. But the thing is, like, there's smaller steps you can take along the way. So just like making connections, learning more. The government is still spending money. So like, and also companies like Altos have like hired a lot of the best like people from like longevity people from universities. And so there's actually open positions, which is kind of strange. Like, and so I think there is actually tons of opportunities. There's actually more work to be done than there are people to do it. And so, and then one other thing is you can also, sometimes you can go really far down the longevity path without saying you're working on longevity. Like just getting really good at like certain aspect of bioinformatics or synthetic biology, and then taking those like tools that you've developed and skills you've developed and then applying them to longevity later. So there's just so many potential paths right now. I don't think if somebody really is passionate about getting into longevity, there's definitely, and you have like some talent, there's definitely things you can do.
Thanks. So I'd like to take a few minutes and just see if there are questions from the audience.
We've done a perfect job.
That's perfect. Okay.
What areas of potential longevity progress do you think are least advanced right now? Like where could we, as a society or as a field, make a lot of progress?
Just a quick one is that I like to joke that everybody thinks gene delivery is somebody else's problem. I always ask every CEO and CSO of every company I talk to, like, so how are you going to deliver that? And they're like, oh, somebody's going to solve that problem because it's so important, but not me. No.
Right. That point's very well taken. There's any number of ideas that if you stipulate the existence of like a effective gene delivery mechanism, you could do anything in biology, and we would have solved a great many problems, especially if you could do it very precisely. The fields within kind of the longevity umbrella that I think are the youngest, that I think have the greatest potential. One is epigenetic reprogramming, and there's a variety of different approaches to this. And I do think that if I were the communications officer for these companies, I'd be doing the same thing. But there's, I think, maybe not an acknowledgement on the part of companies who are basing their claim on epigenetic reprogramming, just how long it's going to take to get a therapy out of that approach. But I do think that real reversal, and we disagree about whether reversal is possible, but real reversal will come from something like that, all right? It is unlikely to come from the small molecule drugs that BioAid is developing. I think we can arrest certain aspects of the aging process, but I don't think we can roll them back with the approaches we're taking right now. So, the kind of nascent, exciting technologies are things that have this potential to reverse. So cell therapies, induced pluripotent stem cell technology, which is a form of reprogramming, right? Again, there's a delivery problem with cell therapies. You have to make the cells go where you want them. Cells have minds of their own. They go basically where they want to go. That's the big one for me that I think is sort of largely unrealized potential, but I think the potential is real.
Yeah, and on my end, I would just ask you not to rule out old school hardcore theoretical physics from ex-Soviet guys, because it's kind of fun. And we kind of, honestly, I do believe that we have a 20% chance that aging is something that can be completely stopped within the next 20 years. This would be my take, but this is like a big promise to fulfill. But there is a chance. I do believe in that.
You mean slow it so much?
So much that it's negligible, right?
so that you're more likely to die of skydiving.
Right, like that.
You know, the skydiving death risk stays constant.
I was thinking skiing on Mars.
Oh my God. I heard about a guy who died skiing just because he fell headfirst into a snowdrift and
Yeah,
no
that
one was
happened at
around.
Heavenly.
I guess it happens a lot.
I mean,
enough that it's like a few times a year, which is a lot more than I feel like.
your drive here might have
Okay.
been more dangerous.
I took part.
So, to make a joke really dumb, like a really dumb one, it seems like the best way to do longevity is to not ride a motorcycle. I'm curious as to, given the kind of bear market that we're in right now, and the fact that the vast majority of capital that's flowing into the longevity space either is actually just from like billionaires' vanity projects, like to a certain extent, to a first order approximation, the second being kind of late stage growth VCs, or early stage growth VCs. How do you think we can change what are the actions that the longevity community can take in order to make a more sustainable financial ecosystem so that we can kind of increase the amount of capital flowing into the ecosystem so that it becomes a realistic thing?
Yeah, I can take that one. It's a weird question. Nobody believes that the drug staging can be created. Still, there's some money that are flowing inside there, and there's chicken neck problem, obviously. So the most of the money would love to flow as soon as the result is demonstrated, and as soon as someone demonstrated the result, he doesn't need anyone's money anymore, anytime in his life. So it's kind of circular argument, and I do not believe, I felt when I started off 5 years ago, at that point of time, I thought that the biggest challenge is to attract more money into the space. Now, after these 5 years, I do not think that space needs lots of money on top of what it has. There's just a technological breakthrough that should be just done. Some things are just some things are just crystallized with time. So I do not see, of course we should, and for our own sake, to save our own lives, we should advocate for a bigger scale of any research that is done in the area. But I do not see it as a bottleneck of the industry right now.
Yeah, I would just add, I take issue with the assertion that the primary source of money is billionaire vanity projects. I just don't think the arithmetic stands up. Altos, which is the $3 billion of the money that you're talking about, is not all from one person, and according to their CEO, they're not a longevity company anyway. But in any case, that's just sort of a barroom debate. I think the best thing we can do for the longevity business is something that a professor of mine in grad school said when I asked him how to, win at science. He said, well, have a great success early on and then continue to succeed. and I think, so I want to go back to that bet that I talked about earlier. The bet of longevity is that there's a small number of mechanisms that cause most disease, and those mechanisms are collectively what we call aging. Some company operating on that bet needs to have a success, however modest. And that when that happens, right, we already have all this enthusiasm based on zero results, right? The moment we have one result, right, it might be BioAge's drug that just had a successful phase 1. It might be Loyal for Dogs, doing a dog lifespan study. But the moment we have one thing that's based on this paradigm that sort of works, the floodgates will truly open, and we will not have to worry about vain billionaires anymore. It'll be, Wall Street, Main Street, everyone will be throwing their money at us. And I believe that day will come, and right soon.
I want to support Chris's argument that my investors, they pray for BioAge success because this will drive our valuation. So this is so we just sit here and wait for our competitors to succeed. This is the only thing we want.
Yeah. I'll add just one more thing, which is it really depends on what you count as longevity work, right? Like if you count everybody who's working on computational biology, if you count everybody who's working on gene delivery, on synthetic biology as working towards the goals that we have, then yeah, it's definitely a huge industry. Although, like if you look at the National Institute of Aging and then NIH, I would definitely love to see a much higher percentage of their grants going towards aging research based on this hypothesis that Chris has mentioned a few times, that aging is the underlying cause of all the diseases that they spend so much money giving out grants for. And that's going to take turnover of personnel in those agencies. And that's actually a program that I'm working with some other longevity orgs to like help place people into those. Like when a job opens up, let's get somebody in there who like believes the Jero hypothesis.
Yeah, just one more thing. And I just want to thank the questioner for giving us this fertile ground for like, it opened up a lot of interesting ideas. And I just wanted to underscore something that Alex said, which was, and this is a theme that comes up on the podcast that I do, Translating Aging, quite a lot, which is there's so much land. This is a colonialist metaphor, so I apologize for it in advance. There's so much land to take that there's no reason to compete over any of the land. So there's companies on longevity biotech, but BioAge never speaks of them as though they're competitors, right? And the idea that a rising tide lifts all boats is just like very baked into the way the industry behaves with respect to its peer companies. Like we all want everyone else to win because like it makes our lives a million. There's a whole other thing about longevity biotech, which is there's a regulatory issue, right? The aging is not recognized as a disease, even within the field. It's controversial how to define it. The FDA certainly does not recognize it as something you could treat. If it did, there'd be practical and logistical barriers to like running a trial that long. And so as people succeed in the clinic, we solve little pieces of the regulatory problem. How do you get a drug that's essentially going to be an aging drug someday through the regulatory hurdle so that it can reach the market? And every time one of our peer companies, we don't really say competitors, as I said, solves one of those problems. And we're an older company now. We're, definitely like, maybe not adults, but we're babysitter age, like we will probably solve some of these problems for other companies who will come after us, and things will be faster for them. And so, but yeah, just there's a lot of love in the room in longevity biotech. Right. It's not like oncology. Those people hate each other.
So you're all welcome, but not to oncology folks.
Looks like we have one
more question. Of course. Let's do one last question, then we'll do announcements real quick.
Okay, so to follow up on this, I understand that once the tide rises, it's everyone's good, but currently it's still kind of a competitive landscape. So who gets the funding? Okay, let me finish. Right, so for me, we're like a pre-seed stage company developing cyclists for neurodegenerative disorders and brain injuries. So if we want to raise, but it's for any kind of early stage company, right? I see that there's like three main things. I actually see that the VC money, there's a difference between people from, be it ex-tech founders having a lot of money, want to live forever, then there are actual more rational biotech VCs who are interested in any kind of new potential in biotech, and then there will be public sources of funding that are like advancing the research. So if I look at those three, what do you think is the most feasible given the current climate, and if there are maybe like recommendations of how to target that? But that's just
I'll try to answer it first. So I believe that it depends on the company that you're building. Like and then there's a certain problem, at least as I can see it, with pure life sciences investors. They pretend to be VCs, but they're not. They're not looking for ventures, but rather like private equity opportunities with 10x something. 10x is still like almost a VC change, but IT investors coming into biotech tend to be more risk-taking, tend to fund more ambitious project, and tend to behave more in a venture way. So they want to make 1,000x on their investment. So it depends. If you have a diploma from Harvard and you can go with the Boston guys easily. Like you will fit all of their check boxes and you will go well. But if you're not, if you're a bit unorthodox in that sense, so yeah, you can go with the tech guys. But again, both ways feasible.
It's a great question. I think that for us and for the friends and colleagues that I have who are doing this, entities with deep backgrounds and a diverse portfolio are, I think, the easiest to deal with.
Mm-hmm.
And I think that like my favorite like funding entity in this regard is like A16Z BioHealth, which was founded by the founding general partner was Vijay Pandey, who was a professor at Stanford, and I think still is. But they bring some kind of tech thinking to the way that they do their investment, but their knowledge of biology is also impressive. But ultimately, what I think is the best about them is they fund people and not companies. So I think if I were to give advice to the young founder, it would be identify all of the respectable houses who really fund founders before they fund the idea the founder is having, and then try to turn yourself into the founder that they want you to be. And it doesn't mean you have to change your stripes. It can mean you assemble a team that then you can bring before them, and they're like, oh yeah, like these 3 people in front of me can do anything. They say they want to do this thing, so here's $10 million. But really what they're doing is they're saying, these people deserve a shot. And I like that approach, because if I can guarantee you one thing about a startup, it's that you'll pivot at some point, right? So like, no plan of battle survives contact with the enemy, so you might as well have good generals.
Might as well throw in my experience here, because I've done the fundraising process a few times actually, and I think the key is really to know your stuff and put a good team together. Like work with people that are really, really amazing, that make you want to work harder because they're working hard. And doing a startup is going to be very hard work. Anybody who thinks that you can have work-life balance when you're doing a startup, especially at the beginning, I'm not a believer in that. But it can be really fun to be on a team that's like so passionate about what they're doing that they just become obsessed with it. And really do your research. Like one of the things that we did at Synthaga, I was on the founding team there. I was the director of science, and I was there for the Series A raise. We went and talked to all of the other people that had tried similar things to us and failed. Like we actually went and talked to them and even made some of them advisors. And like the VCs were actually really surprised that we had done that. Like, because normally people are sort of so obsessed with their own idea that like, I'm right and this is my idea and it's the best idea ever. It's like, no, we had actually gone and talked to other people that had tried what we did and failed, and be like, why did you fail? And like, and then we actually did pivot quite substantially to like.
You should write a book about what you just said. Like, find people who failed doing the thing you tried to do and put them on your advisory board.
It was
That's
awesome.
A good article. I'd retweet that. Yeah.
I'll ask ChatGPT to write it for me.
You should write a tweet.
All right.
All right, thank you, panel. All right, do we have a couple quick announcements? I know we have, okay, great.
Hey everyone, I'm Stephen Hubbard. I am a bioprocess scientist. I work for Eppendorf. We make a bunch of bioreactors for cell and gene therapies. I am also the founder and leader of the Biotech Barbell Club, which is a group on LinkedIn for people who are in our field and also interested in strength training. Strength training is really interesting because of a couple reasons. Obviously, strength is good. I think that some of the most compelling evidence coming out of these biomarker studies is that strength is really predictive of long-term well-being, health span, and lifespan. Actually, the way that they measure it in these studies is with a grip strength analyzer, and I have one. And if anyone would like to get your biomarkers measured, I'll be around. But two, as a biologist, we like experiments, and strength training is an incredible experiment on your own organism. Anyway, so I'm growing this group. It's on LinkedIn, so it's for serious people who are interested in talking about the complexities of the human physical organism and its response to stress. Okay, thank you.
Nice.
Come join us on LinkedIn.
My name is Doug Hirschberger. I work for ADM, and I'm also a co-founder here at the Bay Area Bioinformatics Forum. We have a position available for a LIMS developer/admin. So if you're interested, or if you know anybody who is interested, basically we are doing strain development to produce biomolecules that are used for commodity chemicals that will replace things that are made nowadays by oil. So the strain development process, we're looking for LIMS to help drive that process of synthetic biology, from low throughput to high throughput synthetic biology, and then fermentation and tracking the fermentation results all the way as we go into production. So we're looking for somebody to own the process of developing our LIMS to capture all that data and integrate it and put it all together. So if you're interested, tinyurl.com, ADM LIMS, or just drop me an email or a resume, and I look forward to hearing from you. Thanks.
Hi, my name is Dashra Trivatsa, and unless you're brown, you can't pronounce it. Please just call me Dash. I'm a startup bro, and therefore I must hawk my wares at every possible opportunity. My company builds an operating system for the computer architecture of a biology lab. And right now we're doing kind of user testing. If you want to try out new LIMS systems, if you want to try out visual data flow pipeline builders, if you want to try out advanced scheduling algorithms, hit me up. I'll give you a build and watch you be confused on it and then fix your bugs. That's what I'm looking for.
Right on. All right, well, thank you, all of our speakers and panelists. Thank you so much, CircuitLaunch, for having us. And
just a quick one. Anyone, you guys can hang around for a little bit if you guys want to talk. If anyone's interested in seeing our lab facility or taking a tour of the facility, I will do one in the next 5 minutes. So just meet me in the back, and we can talk. And again, thanks you all for coming. So cheers.
Thanks, everybody. That was great, you guys. That was a lot of fun.
Thank you,
Chris.
Chris.
That was fun.
So the way it works is someone squeeze it.
Thank you so much.
It records the highest value.
First panel discussion.
Right, and then
I know. We'll do the last.
start. It resets itself.